botulism. And so as you can see, this is the only picture I have. It's similar in appearance. It's another, like I said, another species of that genus, and it causes. The one we looked at was mainly a bacteria that caused food poisoning, and in this case, it's a little bit different. There is some food poisoning possible with Clostridium perfringens, which is what we're going to talk about today. But it's gonna be a little bit only. But it mainly is of concern because it causes myonecrosis. And if you put these words together, necrosis is always the cell death, and in this case it's muscle cell, so it affects muscles.
Myo is muscle.
Yeah, and then the gas is basically, we know it's anaerobic because it's the same genus, and we had talked about how they're anaerobic. So they're producing a gas in most instances, and so you have kind of like a gas formation under the skin is something that you can see, and it's also therefore called anaerobic cellulitis. So it's a change on your skin, and you can see it by bubbles. And I think I have a picture that shows you. So it's the same. It's pretty close to the one we talked about last time, and I want to keep those together. And there's going to be one short discussion at the end about another one called Clostridium difficilis, and that's a third one I was just going to introduce real quickly because somebody sent me an interesting article about it a week ago, and he said maybe that's something you want to mention, and I think it's going to be really interesting. So that's at the end. So I was going to start with a case file, and it's interesting that a lot of times in nurses, everybody who ever wants to enter that profession is going to see a lot of that and be really, what are you going to be? Well, at least nervous, because I remember when I took physiology, and in Germany you can take, or in our system, I could take physiology in the medical department. So I didn't have to take I was studying biology, general biology, but I took physiology, human physiology, and I think anatomy in the medical department. So they were always talking about case files. And what appeared, and which is the same thing here, is that a lot of times, you have, if you understand the physiology and if you understand the disease, it's very simple. It seems very simple, but you get a lot of patients. A lot of times you get somebody off the park bench at 3 o'clock in the morning and you don't know what they've done. And so there's so many complicating factors in there. And I just say that because it was always interesting to me to try to somehow help them at the same time. You don't know what's in their body, and there's certain ways doctors try to treat, but then that's impossible because of the alcohol, or there's lots of compromising factors. So this case is the same. This was the same kind of case. There was a 39-year-old man. He had severe pain in the upper shoulder and chest, and he, over the course of, he came in, they looked at him, and after a while he admitted to injecting drugs on a regular basis, but also having it done just recently with a combination of drug, water, and then with a non-sterile needle. So you have already, you have the problems of, maybe a compromised immune system. You have problems of, you don't know what else is going on with the rest of his organs, how well they function, the liver, et cetera, et cetera. So they don't really. Initially, they did a CT scan and they saw severe inflammation in these areas, and it was spreading towards the arm. And so there was basically tissue necrosis, so tissue is starting to die. And so they took tissue and fluid samples and looked at it, and they said, well, they found a mixed infection. So that's one of the problems where you don't just find one organism, you might find multiple. And also how they interact. For example, you could have a Clostridium in there and it needs anaerobic condition. So you would say, if I expose it to air, it might not be able to survive. But then you have other organisms in there that use up all the oxygen. So they together, they use up all the oxygen, make it easier for Clostridium to spread into other areas where there's no air. So it gets pretty complicated. So what they found is it's a clostridial myonecrosis caused by perfringens. And usually that's a non-invasive species or bacteria, and it enters through skin piercing injuries, lacerations, and then just like in the food poisoning, you have spores again that germinate. Remember in botulism we said that you actually do have that organism in you, but the spores are the ones that cause the problem, and or the spores are the one that survive the heating, and then they develop into vegetative cells. And that's the same thing here. So they germinate in that damaged tissue, and then they create inflammation with their toxins they produce that creates necrosis. And they're able to multiply, so they're spreading, and whatever else. There's enzymes being produced, but also gases, and that creates those gas bubbles. And you can see that here on in this picture under the skin. Maybe I'll leave this out for this daily disease. And then it spreads into healthy tissue, so you're trying to somehow get it under control. But obviously, if the bacteria is happy in its environment, no air, lots of nutrients, it can spread fast. And in this case, the patient almost died because he had an additional infection with Clostridium difficile, and I'll talk about that one at the end. So I already showed you this from the last time. It's just basically you have gram-positive rods. They're spore formers, and they're obligate anaerobes, and we talked about what that means. So that's where they belong, and all three organisms we've then talked about for botulism and gas gangrene, and then for diarrhea, which is severe with those Clostridium difficile cases. I've showed you this before, so it's just to go back to the genus Clostridium, that they're gram-positive spore-forming rods. They produce them only under anaerobic conditions, and then they produce organic acids, alcohols, or what causes the damage. And so wound infection, tissue infections, also food intoxications. So generally you find it again, like the other one, in soil and water, but you also find it in the intestines, vagina. So you do find them in humans. It's just like with the one with Clostridium botulinum, you find it also in the human. It's just in a different environment. It's under control in your intestines. It's different when it enters a wound through, for example, an injection with a dirty needle. So it produces several enzymes that cause tissue destruction. There's one called alpha toxin, causes red blood cell rupture, edema, and tissue damage. And so those are the causes. We've talked about these already: myonecrosis, gas gangrene, and anaerobic cellulitis. But it can also cause food poisoning, less so than all the other possibilities. And you can see here, so this would be a muscle, these would be muscle cells, and you can see how the bacteria is in between those muscle cells in these spaces, or these muscle fibers, and these bacteria are in between, and they're producing gas, so they're basically they're increasing the spaces between those fibers, and you see most of that coming to the surface, so basically under the skin. And what also can happen if you have a compound fracture, meaning you have a fracture where you actually where say the bone gets out of the, or what do you call it, protrudes out of the skin, and you would then get infections into you can get infections into your, say it's on a leg, for example, and so you have necrotic necrosis, which means you have a blackening of the tissue, and you have bubbles forming under the skin or on the skin from gas formation. So factors are surgical incisions, like I said, compound fractures, puncture wounds, gunshot wounds, and a lot of times when you think about it, initially I would think, well, where do you really find these areas where you have no oxygen in those wounds? But you really mainly from interrupting the blood supply through those injuries is why those anaerobic organisms are able to grow pretty well in there, or their spores do. And then also, as I said, there's an addition of aerobic microbes that come in say a fracture, and so they use up all the oxygen, and so the conditions for Clostridium are getting better. So you'd have to clean the wounds, number one. In this case of the 39-year-old, a lot of the tissue has to be removed, so you really try to get the tissue that's infected with Clostridium away, so it can't spread. You want to remove that to eliminate the anaerobic environment, and then you give antibiotics. And the other thing they do is hyperbaric chambers, and that's just because you apply a high concentration of oxygen, and you hope that infiltrates enough into the tissue that makes the sporidium growth or decline or decrease. So that's one of the things that they show in the book, I think, the chamber. So the problem with the 39-year-old is that he also had this Clostridium difficile infection, and what happens here a lot of times is they're normal residents of the colon, but usually in low numbers. And what happens is a lot of times when you give antibiotics, which in his case happened, they kind of they survive initially, and a lot of times those broad spectrum antibiotics kill a lot of the other bacteria, and then difficile is the one to take advantage of it, overgrow the other ones, and then basically become super infections. And they produce enterotoxins that damage the intestines and are a major cause of diarrhea in hospitals, and they're increasingly common in community-acquired diarrhea. But the main thing is, and that's where I got this article, is that they see it more and more, and they're trying to figure out how it spreads, especially in hospital situations. So it's kind of a concern right now. And the interesting thing is, we'll get to that after this slide. So most cases respond, or the mild cases respond to fluid and electrolyte replacement and withdrawal of the antimicrobials. So you just take them off of the antibiotic. You try to give electrolytes, try to give fluids, and then hopefully the colon recovers and doesn't look like in this case where you have growth on the walls. So those are images of what the difference of a healthy colon to a infected colon looks like. But the other thing what they do is, and that's a big discussion, and I was just going to tell you about it because I think it's interesting. So what they're talking about is replacement cultures. And I heard on the radio this weekend that more and more microbiologists start thinking, or they have this idea of our microbes being really almost like our genes. So if they compare family members, for example, or generations in families, we have very similar microbial cultures or microbes in us. And so they were talking about how that is almost like a genetic fingerprint. And it could more and more become something that doctors do, where they say, well, we need to know what you have, what kind of, maybe we'll focus on 10, whatever, 20, but we need to know, what your, I guess, microbial flora is composed of, and then we can tell you how, maybe certain risk factors for you, things like that. So that becomes more and more important. And what they say is for replacement cultures is, because the precautions to prevent the spread in hospitals is currently not really successful. So what they here, and that's what I was talking about. So the infections have risen in numbers and severity. They're actually estimating 14,000 mortalities or deaths per year, and that's in the United States. And so there's this idea of a fecal transplant, and that just basically means your flora is destroyed. You're gonna get it from a family member or from a donor, and obviously that it's something you first of all probably won't forget. That's why I bring it up. And then it's interesting because we'll go through this, and there's only one more slide, but it's interesting because if you ask anybody, nobody wants to even think about it. But at the same time, it seems like it's a real solution. And there were a couple of articles about this, and so it's not a completely new technique either. They've actually used it in China. I don't know how long ago, and I don't know how either. But they said, for example, if you do like these big studies and you include all of this, you have 273 patients. That's patients from all kinds of other or different, with all kinds of diseases, but are always related to your intestinal flora, I guess. And they a lot of times saw a resolution of their disease, and also didn't report any adverse effects, because you really are. There's nothing. I'm not going to say anything more. I'm just saying the big advantage is it's microbial matter. It's nothing else. And so these are the methods that you can apply it to, and I think colonoscopy is the one that is mostly used. And it's so far not I don't know how all these work. And so what basically happens is you get a, is that in the next slide? You really take a sample from a donor, which it's not a common practice because you have to first determine who can be a donor, have they taken antibiotics themselves, do they have any other, that's going to be a long ways, but it's been practiced before in cases where there was no other plausible solution. And one other thing that's important is if you have that kind of the Clostridial diarrhea, the problem is it comes back a lot of times. So people are pretty desperate to find a cure. Even if they give antibiotics, even if they get rid of it, they get it later on again in a few weeks or months. And so they want to totally get rid of it. And basically the idea is you give somebody healthy microbes that immediately deal with, you basically flush out whatever you have to, and then you give them a new flora from a donor. And so this is just how that would work, and they would probably, or most likely, your intestines would be flushed with this sample that's diluted, and then.
So is it usually a family member?
I don't know what they've used so far. But they've done a lot of studies, and it seems to be. I mean, in that article, it sounds like this is the newest best thing ever, and people just have to get over it. that's. I should show you. Oh, I can show you. I have somebody sent me a copy. And then, if you think of acceptance, I mean, what would you expect, or what would you yourself say? I mean, if you have to make that decision, it's hard to do without having the actual disease, but. Would you be absolutely opposed to it and say, no, you got to do anything else, but I'm not. it probably depends mainly, I would say it mainly depends on the application, right? I mean, you would say,
I would like the naso.
yeah.
Yeah.
Yeah, if you're completely passed out, probably. I mean, I'm not gonna give you my opinion, but what they say is that so those with recurring infections of Clostridium difficile would 97% are willing to undergo another. So they've got one and they do it again, and then 53, so more than half would actually take this therapy before taking antibiotics. So what they're now trying to do, which is like more of a,
This is all in the US?
yeah. It really is. It's probably something you don't, and that's why I wanted to bring it up too. It's something that you don't hear that much about.
Well, if you don't think about what it is, it's probably not that bad.
Yeah. Well, you know, and then you can go back and think. Obviously, our big or the main problem with it really is I mean, it has a biological. There's a biological reason for us to be so.
Grossed out.
Yeah. And it's because they're, you know, it's, you know, for dogs it's a different story. But it totally depends on how we can deal with it. So there's a reason for it, and so it's not just that you grew up being taught that that's not right or whatever. But what they're trying to do now is they're actually trying to basically artificially produce what they call a probiotic soup, and they basically would artificially produce something that you know, contains about 20, say, enteric bacteria, and then that would be given to you.
Instead of.
Yeah, and so that obviously sounds
It's better.
a lot better.
You're not
So that was
swirling
to
and filtering through gauze.
so that was about Clostridium.
I don't want to do that. But then you'll have all the fear. I would go for the artificial pieces.
You have to wait. I don't think they're there yet, but it shouldn't be too hard. What do we have left? 35, 40 minutes.
It's just good, right?
So I'll try to make this I really will try to make this, and you're gonna disagree after this, as painless as possible. And I think the reason for it too is I don't want you to leave this class and say, and then somebody else asks you and they'd say, how come you don't know anything about it? Or how come you're not so sure about say metabolism and how this all works? But really it doesn't, in my you know, in my experience or also in my opinion, it wouldn't really matter. It doesn't matter as much. You know, it's basic. It's biochemistry. It's basic, or not basic, but it's biology, where you would have like an advanced biology class, and you would learn, like some of you already did. You'd learn about metabolism and fermentation and aerobic respiration, and then photosynthesis. And we're just, all it is really to give you an idea of what kind of energy production there is, or what kind of metabolism, because there was a question today in the test, hopefully lots of people got that right, but metabolism is really the summary of it all, that we always talk about all the different things that happen on the cell membrane and in the cell and the ribosomes and the basic, it's biochemistry, it's basic, or not basic, but it's biology, where you would have like an advanced biology class, and you would learn, like some of you already did, you'd learn about metabolism and fermentation and aerobic respiration and then photosynthesis. And we're just, all it is really to give you an idea of what kind of energy production there is, or what kind of metabolism, because there was a question today in the test, hopefully lots of people got that right, but metabolism is really the summary of it all, that we always talk about all the different things that happen on the cell membrane and in the cell and the ribosomes and the what everybody does and or what every organelle does and all that. And in terms of microorganisms, all of them have to have a metabolism to, they're constantly taking in nutrients and they're trying to keep homeostasis with it, and they're trying to get rid of their waste. So that's really what this is about. And I'll try to point out, just so you know the major pathways they can use, because you have aerobic bacteria, you have some that only grow in oxygen deprived, like the Clostridium, the organisms under the genus Clostridia, and then you have other ones that ferment. You have yeasts that we use for fermentation and for alcohol production, so that goes more under the kingdom of fungi. But there's all these microorganisms have different ways of metabolizing energy or metabolizing substrate and creating energy, is what I should say. And so, just so you have an idea of what the possibilities are, I'll do that. And I'm not going to go into, if you go through glycolysis, there's probably, I don't know, 10 steps, 8 steps, and you have all these intermediate substrates starting with glucose. And I can't remember if it ever, in all the the work I did, I never had to use it. I mean, it's good to know what they do, but you have to really be researching maybe the pathways of metabolism in an organism to actually have to remember and really look at all the compounds. So I hope I can just point out the most important enzymes and products in the process, and give you an idea of what they go through under different categories, and not overwhelm you with too many formulas. So energy is obviously needed to do work and to cause change in the cell. That's the number one. So the energy has to come from somewhere, and you've got all these different sources of energy. You, for example, have light. You have thermal energy, mechanical energy. There's energy in the nucleus that we're using for in the energy producing plants and power plants in nuclear power. And then you have chemical energy that's present in the bonds of molecules. And that's really all we're going to talk about. So the cell, because the cell is fragile, it's a small fragile structure, wouldn't be able to handle a lot of the energies that come from these other sources. So the energy that's in the bond of a molecule is really what it can use. And just try to remember that and try to make that or make sense of that, because we've talked about before. You have like, say, glucose, and it's a molecule that harvests, or not harvests, but that has energy in its bonds, and through certain reactions that energy can be freed. And so if the plants produce certain compounds through capturing the energy of light, which is photosynthesis, we eat either plant-eating animals or plants, or anyway, we eat structures, and we can capture that energy from those compounds they produce. So they produce cellulose or starch, and we can break that down, and we can extract the energy out of those molecules. So that's why they're the chemical. And so mainly, and we talked about those before, we have anabolism and catabolism. And so if we're building, usually it consumes energy. So we have to put in energy, and we can create a product. So those are inorganic reactions, and exergonic reactions are the ones that are actually releasing energy. So those are the ones that usually you get by splitting, by releasing the energy from an energy-rich compound such as glucose. And then this energy, a lot of times, and we talked about that before, so the body is, or the cell is constantly extracting or getting energy from substrates, but it's also using it immediately to build. So it's not just storing it in a compartment in a battery and using that up later. It's actually using that energy immediately to build. So the catabolism and anabolism are always going hand in hand.
Why does it say it's temporarily stored in high energy?
Well, because it's and we get to that. So there is kind of like a storage unit, but basically instead of, you know, sometimes it has to be transferred. Remember in the enzyme, the energy, or maybe I don't bring up the enzyme yet because it's going to complicate things. But so the energy is stored in a. You know, say for example, something here is used or you produce energy by going through this reaction that releases energy in the cytoplasm, but it's needed somewhere else in the cell. There is a way, and that's what we're going to talk about. There are high energy phosphate molecules, and that's going to be ATP that does store that energy. But it's basically those bonds are not very stable, so it wants to, and I'll show you in a second why, and it wants to get rid of some of those phosphate groups, and that releases the energy. So it's basically a transfer of energy from these substrates to a compound that can pick up phosphates, release phosphates, so pick up or use up energy and give energy, or that energy. But it's basically, those bonds are not very stable, so it wants to, and I'll show you in a second why, and it wants to get rid of some of those phosphate groups, and that releases the energy. So it's basically a transfer of energy from these substrates to a compound that can pick up phosphates, release phosphates, so pick up or use up energy and give energy. So it's just and ATP is one of those cofactors we talked about. It's basically one of those factors that help in enzymatic reactions by transferring. Remember we had like one of those cofactors that was transferring groups or you know, a carboxyl group, or so that's what ATP does. It transfers electrons, but you'll get a better idea of that in a little bit. So do you remember what this is? This is an orbital. This is a proton. This is an electron. Does anybody remember? Because this is the most important one we'll talk about today. What is that?
Hydrogen.
So this is important because the way the energy is usually stored is in electrons, and so if we say the energy is in a chemical bond or in a molecule, that's hydrogen is really the one that basically hydrogen and its electron is basically what's transferred in all these reactions. So if you have glucose, what really happens is you're basically taking off these electrons along with the proton, which is H+. So you always have like basically, because this is, so when it loses its electron, it's H+, but it's H++ an electron. So that's really what a lot of times is produced, and then that can help to form ATP. So that energy goes into here, and that one can then give the energy to something else, to some other process, to building or any other process that, you know, it's for example, for muscle fibers to actually have, you know, your muscles contract, you need ATP for anything. But to not go too far, so in general, we've talked about oxidation, and that's oxidation and reduction, and that's why that's always important, because those redox reactions are what what's really important in this process of making ATP or gaining energy out of this process. How these redox reactions are really important to get the energy out of an energy-rich compound like glucose. So you always have an oxidation going on. So if you look at this whole process, then that would be respiration, aerobic respiration. So there's oxygen, and I don't know if you remember, but that's I gave you this formula before, and if you convert it, if you turn it around and you put, instead of energy, sun energy in here, then it would be photosynthesis. So this is just, I told you, I wrote this formula down before. It's basically the building. If you look at it from this way and you look at it in this direction, it's the sun energy and water and carbon dioxide builds these simple sugars and releases oxygen. And on the other hand, what we do in respiration is use these simple sugars and oxygen to produce energy for ourselves. And so that's the main formula behind it. And the redox pair in this case, you always find, in this case it's hydrogen oxidation to this form and then reduction of oxygen, and it's all contained in water here. So and I think it makes more sense when we go along. Just remember, there's always a pair. There's always a redox pair involved in these reactions, and this whole process is a long process, and it has three distinct phases going from here to here. But in the end, those are your end products, and there's elements oxidized or molecules oxidized and some reduced. And the energy that's being released in this process can be captured to phosphorylate ADP, so it's a diphosphate or 2-phosphate, to an ATP, which is 3-phosphates or other compounds. And like I said, that's going to be making more sense once you see it a little bit further. But if you look at this again, and don't worry about the written part, so you have, if you look at the y-axis, you have energy level of the compound. So it's low here and it's high. So any compound that you see here, energy level is high here, low here. And then you have the progress of extraction of energy over time on the x-axis. And so if you look at glucose, this is what you start with. And I want to make sure you understand, it's not just glucose. So it's not just like glucose is the only molecule we can use. I mean, there's fructose, there's other simple sugars that can also go into this cycle and can be used, but there can also be, fats, for example, that we burn for energy, and they come in somewhere else, but it's usually always explained by using glucose. So you have a simple sugar and it's energy rich, so it's high up here. And it's basically through a lot of reactions dropped down and converted into carbon dioxide and water, which we just saw in this one. So you have carbon dioxide and water. So that's the product of it, the end product. And along the way, basically you have these reactions and hydrogen ions, so H plus and electrons are constantly given off, and they enable the formation of ATP. So this energy that comes out of this rich energy, high energy compound is released step by step by electrons and hydrogen ions, and it creates ATP along the way. And then you have, so ATP is really the final product, or not the final product, but it's ATP is what you would gain from all these, from this pathway, but along the way they're electron and proton carriers. So that just means if you have an electron given off, you remember all this time, so there's a hydrogen ion and there's an electron coming off, and it changes, and you slowly lose or there are parts of enzyme that are able to accept electrons and protons. And there's NAD, FAD, NADPH, coenzyme E. Just maybe remember a couple of those. And so you can see here, for example, if you have NAD plus, this is what it looks like. And now you don't have to know the exact structure. I mean, you always have a sugar, and it looks similar, a base. It looks similar to what you've seen before in the macromolecules we talked about. But really what it can do is it can accept these electrons and protons and then carry those and eventually give them off to produce ATP. So they're along the way from going from glucose, which is a C6. You can see that here, or so you have a C6 and you go down to a 1 carbon. So you go all the way from 6 down to 1. Along the way, you're gaining energy, and those electron and proton carriers accept that energy and eventually help to make ATP, and that's really the form you need. That's back to reduction and oxidation. So if a molecule has been reduced during a reaction, in a reduction, you always gain. So the only two possibilities are, I mean, I don't know how you explain it to yourself, but I would say in reductions you always gain, and it's usually electrons and hydrogen. So it's important to remember those two always go together. Protons and so D would be the right answer for this.
What did you say?
D. So before I show you the 3 processes, and I'll try to go through those quickly, not worry too much about the detail. ATP is so important that they call it the metabolic currency. So that's really, that's where the energy is stored temporarily, and you need ATPs in your muscles all the time. You need ATP for any kind of energy using process. And so you have a ribose, which is a 5-carbon sugar. Again, you have a hydrogenous base, adenine in this case. It's up here, and then so it doesn't look that different from this compound. You have the ribose, adenine, and then in this case you have a different group attached to the ribose, but this one is more important. And what's really important here is that you have these 2 components, and then you have these phosphate groups. You have so you have AMP, which would be monophosphate, so you would have 1 phosphate in AMP, and then you have ADP and ATP. So those are, and it basically means, if you would look at them, we'd either have 1 phosphate group, 2 or 3. And they have, as you can see, they have really, and that's just a detail, but they have a high negative charge. And so if you think about like this chain of phosphate groups, and they all have negative charge, they kind of want to, they're pulling away from each other. That means they're unstable. So this compound is unstable, and because you have all these negative charges, and the more you have, the more unstable it is. So if you have these 3 negatives all together in this phosphate tail, basically, they want to really pull apart. So it's the energy that's in this bond is released easily. So that's why it's such a great carrier of, or such a great cofactor, because you really do. The energy, or that phosphate group, is easily attached but also easily given off, and it takes energy to put a phosphate group on it, and it releases energy if you get rid of a phosphate group. So in a regular active cell, you have a constant utilization and replenishment. So you're constantly making ATPs, forming ATP, then going back to ADP or AMP, and then the reverse. So it's constantly, you got all three of them down here. There's a constant going back and forth. And if you think, I mean, it blows your mind if you look at E. coli, a pretty simple bacteria, has about 2.5 million ATPs used in a second. If you imagine in 1 second, that's what's going on in a cell, and that's like a one single cell. So you don't even want to start counting on what's going on in your own body. And I don't know how how anybody comes up with those numbers and how they figure this out, but it's pretty amazing how much detail they have on how we metabolize substrate and make energy, and how ATP is constantly replenished and does what it does in every single cell. So this should give you a better idea. Remember how we have, like, we always talked about this apoprotein, the big part of the protein, and then it has like an active site, and it a lot of times has like coenzymes attached to it that are helping to transform or not transform, but to transport and to bring groups from one substrate to the other. And in this case, the big protein is called hexokinase, which you don't have to remember, but that's the protein, and you have glucose. And so in this first step, for example, which is actually uses energy, it's just the first step of basically prepping glucose to go into this metabolic pathway. You actually add a phosphate group to it. So this costs energy. So the whole process starts with actually putting energy into the system. But anyway, we're not going to go through all the steps, but this happens to be step one. So you go from ATP to ADP. So you give energy to the whole thing, and you attach this phosphate group to glucose. And so this is just basically a picture showing you how this works in how this is enzymatically done, going from glucose to glucose 6-phosphate, and how ATP here with the 3 phosphate groups can basically transfer one of those phosphates onto glucose, and so you have glucose 6-phosphate, which is where on that this just describes the position of the phosphate, and then you have leftover ADP, and then that can get is a phosphate group later on, again to become ATP. So if you have, this would be a reaction that costs energy, and on the opposite, you could have the same kind of reaction where the phosphate group is transferred onto ADP, and that I'll show you is one of the main reactions at the end of it all, because in the end you want a lot of ATPs. That's what you need. But that's at the end of this whole all these different pathways. And so the formation of ATP really happens in 3 ways. And if you want to start at the bottom, which I'm not going to talk about today, but photosynthesis is one way to form ATP. So that would happen in a lot of the plants. In plants happen, you would see that formation of energy, or formation of a lot of the substrates, a lot of the structures they're building by using sunlight. And then there's 2 other ways of forming ATP, and there's the substrate level phosphorylation. So basically it just means you have, just like here, you have a substrate like, say, glucose 6-phosphate that has a phosphate attached, but it can go the other way, where that's here, and you have ADP, and through an enzymatic reaction, basically the reverse that we just seen, you would get the substrate without the phosphate group, and then the phosphate attached to ADP, which makes it ATP. So that's one of them, and that happens in the process. So sometimes substrates give away a phosphate to ADP to produce ATP, and that's what you call substrate level phosphorylation. And remember, this is all to form ATP. And the other one that's important, and that's all beside, with the exception of photosynthesis. The other one that's really important that happens, or that gives the biggest gain in ATP, is the oxidative phosphorylation. And so that just means there's a series of reactions during respiration, and we'll get into how that looks. And that series of reactions produces or enables a lot of ATP to be produced through ADP and a phosphate, and because it doesn't in this case, and you'll see why the phosphate doesn't come from a substrate. It's not a substrate level phosphorylation, it's oxidative phosphorylation. This is the most important one in terms of how much ATP is produced. So basically what this is explaining is you have catabolic and anabolic reactions, and most of the energy you're going to gain is from using fuels, just like glucose or fructose or maybe a fat, and you go through a series of pathways to end up with energy. So just think of, a regular cell gets glucose. What does it do to actually make ATPs out of it? And it's a long process. It's not just, it can't just take, glucose is a high energy substrate, and it somehow needs to become a single carbon dioxide, and water. And how does it get from up there to down there? And how is the energy from this energy contained in glucose transferred? How is it transferred, number one, and then how does it help to produce ATP, which then can be used for everything else? And so the three steps that it usually goes through in under aerobic conditions in any of our cells, we have oxygen, so we have aerobic respiration or metabolism. You would go through glycolysis, and then the Krebs cycle, and then the respiratory chain, electron transport. And this, in the end, really, these are all important, but this, in the end, is where you have, and I should almost show you that you don't wait too long for the solution, but this is where you get the ATPs. Let's go through all the ugly. So in the end, if you have a cell like ours, or a bacterial cell, you have this ATP synthase, is what it's called. So it synthesizes ATP. And don't worry about how it does it, but it creates ATP out of ADP and a phosphate. So it produces this, and this is the end product of all this, is basically, and I'll just show you so you don't have to wait forever, but this ATP synthase is what makes the ATP, and we get to how that's done by taking out by using up the electrons from a high energy compound like glucose. So just remember those three. You have glycolysis, a Krebs cycle, and a respiratory chain. And glycolysis would be, we talked about just so you have an idea, we talked about this step by step, single pathway. There's an enzyme, there's always enzymes involved to the substrate, and then you go down this pathway and it's a single straight step by step. There's all these enzymes in between, and you end up with, like, say, another compound here. That's your intermediate product. And then obviously the Krebs cycle is a cycle, so you kind of whatever comes out here cycles, and you produce, or you're able to that enables the cell to produce some of those to take up some of those electrons and hydrogens, and then use them later in this final production of ATP. So you start off with a high, in glycolysis, you start off with a high energy substrate like glucose. You go through the glycolysis, through a Krebs cycle, and then in the end, in the mitochondria, I don't know if you remember that, but that's where all this happens. You get most of your energy in form of production of ATP.
Like the whole thing from the mitochondria, or just the?
No, the glycolysis usually happens in the cytoplasm, and I have that listed. The main difference is, and that's in our case, because as you know, prokaryotes like the bacteria don't have those organelles, so they do everything in the cytoplasm, and then this final important transport of electrons and production of ATP happens at their membrane. So the eukaryotic cells use their mitochondrial membrane to do this, and in bacteria it's their regular cell membrane, because they don't have mitochondria. So that's the main difference. They don't do that in that's the main difference, the location of the respiratory chain.
Is that more evidence of?
No, I mean that would almost be the opposite, where you would say, well, one evidence out of this would probably be, or it supplies or creates evidence to see the resemblance between prokaryotic and eukaryotic cells. Because you would say, if a bacteria can do pretty much exactly what we do, just over a different membrane, that's amazing. I mean, how come our cells that are so different in terms of the size and all their organelles aren't using a completely different way to make energy? They're using exactly the same, depending on what bacteria you look at. I mean, there's some, obviously the ones that use, maybe that produce methane, or that use, there's lots of bacteria that live under different conditions. They do it differently, but it's actually not that different, and you'll see why. I mean, but it does give you, or it does make it more likely that we have the prokaryotic and the eukaryotic cells have evolved, or the eukaryotic cells have evolved from the prokaryotic cells. And it wasn't just both of those cells developing independent from each other and by accident using the same kind of metabolism, which is really, that's at the basis of everything. So it would be a big coincidence. So these are the three processes we're going to talk about, and I already showed you a little bit of this. So the aerobic respiration, which is what we do in our cells, means we need oxygen. So any oxygen breathing animal, for example, you can pretty much guess that they're going to go through glycolysis, or their glucose, their energy production is going to go through this process of glycolysis, Krebs cycle, respiratory chain. And basically the oxygen, and you'll see that too, the oxygen is really important. If you go back here, because it's the final electron acceptor, and I didn't say that yet. You'll see that when we get there, but this is where where the electrons in the end up with the oxygen to produce and the protons. So basically, with the hydrogen, the positive hydrogen. So you basically the oxygen we breathe is used to accept these electrons at the end, and you produce water. So in any animal that's breathing oxygen, that's the purpose of oxygen. That's what we use oxygen for. And so if you have anaerobic respiration, in some bacteria, for example, you can go through the glycolysis just like those. The first step's the same, second step's the same, and then the only difference is that you don't have oxygen available because it's anaerobic respiration, so no oxygen is available. So the only difference is there's a different final electron acceptor. So you don't have oxygen. There's not oxygen that you have to accept the electrons and produce H2O or water, but you have compounds like, or you have oxygen containing ions. And they have, for example, they have carbonate. This what else was there? Carbonate, SO2 SO4 there's a sulfate, carbonate, and there's one more. Can't remember. They're gonna come up next. But they have basically oxygen-containing ions that they use as final, and that's really the only difference, as final electron acceptor. So always anaerobe, remember in that respiration, in the aerobic respiration, you have oxygen that stands all the way at the bottom. So you start with glucose, and you end up with all that transferred to oxygen. And that's why it's aerobic respiration. In anaerobic respiration, you have ions that contain oxygen, but not oxygen itself. And then in fermentation, which is a third process, you basically only go through glycolysis, and then you have some compounds, organic compounds, that accept the electrons. So you don't go very far in that whole process. And so the outcome, the ATPs you actually can gain from these different ones is pretty high in those two, but very low in this one. So fermentation doesn't produce you a lot. It's just you don't have any choice, and there's advantages of doing it this way, but it's for example, that they don't depend on oxygen like we do, but it doesn't produce a lot of ATP. So they calculate under ideal conditions you probably get about 38 ATPs out of this whole process, including those 3 processes, and here you get 2. So you don't gain a lot of energy, but the advantage is that you don't need oxygen. You can do it quickly. Some of those organisms do it really quickly, and so there's other reasons for fermentation. But it's just also it might be part of occupying a niche. There are organisms who are able to live without oxygen, and so they can thrive where a lot of the ones that depend on aerobic respiration can't. So those are the 3, and just to point out again, I mean, if you remember the basic concept, you start in those respiration. So remember, respiration meaning you start off with glycolysis or with glucose. So you start off with a C6 substrate, and after glycolysis you have 2 3-carbon compounds. So you basically split it in half, and then you go through the Krebs cycle, and depending on what you do, you either have oxygen as your final electron acceptor, or non-oxygen electron acceptors, like these ions, SO4, 2 minus, NO3 minus, nitrate is one of them, carbonate, and sulfate. So either those, that happens in some bacteria, and then in the last, in the fermentation, you basically stop here, right up here after glycolysis, and you go into 2 or different pathways. There's a lot more than these, but some of them we know is you produce, the organism produces ethanol. So these are these organic compounds are the electron acceptors, not oxygen containing ions, but organic compounds, and so those produce lactic acid, alcohol, a lot of the other acids, acetic acid, so vinegar. There's a lot of different acids and alcohols produced by organisms that use fermentation. So what are we now? 14. Yeah, I'll have to cover that. That was actually I was going to try to finish this. It took a little bit longer for the exam, so we'll just do some of this tomorrow. In addition to just looking at slides, and hopefully it'll be slow enough that it kind of makes sense. I know it's tough if you haven't ever looked at it before. And then we'll go away from this and just talk about how to control bacteria, what disease they cause, things like that. So it's going to get